Welcome back to the Deep Dive. Today, we are performing what is, I think, a crucial autopsy on one of the biggest medical headlines of the decade, the 2022 pig heart transplant into David Bennett Sr.
It was a massive story.
Huge. And, when we talk about organ transplantation, we're talking about this incredible life-saving potential. But then there's the other side of it, the stark reality of the organ shortage.
Exactly. For so many patients, the average wait is 3, 4, even 5 years.
And they just run out of time long before a human donor is ever available.
And that urgency, that's really why the focus shifted so radically to xenotransplantation using animal organs. For patients with terminal heart failure, this is really the only emerging new option. The mechanical assist devices, they have just such high complication rates.
Right. And the moment this all moved from theory to a genuine clinical reality was that transplant in early 2022. It was a compassionate use case, but that pig-to-human heart, it functioned.
It did. It was beating for over 45 days. It was a massive proof of principle.
The absolute definition of it.
45 days. That's the point where they proved they could, in essence, hack the immediate rejection system. But the real lesson, the painful lesson for the whole scientific community, wasn't that initial success. It was the failure at 2 months.
Okay, let's unpack this. That's our mission today, really, a forensic analysis of that 2-month outcome. We need to move past the headlines and really understand the complex biological, the immunological factors, everything from specific pig viruses to, standard hospital therapies, that ultimately led to the failure.
And what we can learn from it.
Exactly. What specific hurdles are now dictating the design of the next generation of human clinical trials?
Well, to really understand the failure, we have to start with the blueprint of the donor organ itself.
Right.
The pig is the species of choice, mostly due to its size and a growing ethical acceptance. But this wasn't just any pig heart.
No.
It came from a genetically multi-modified clone donor pig. It had 10 specific genetic modifications.
10 modifications. I mean, that sounds less like a pig and more like a carefully engineered biological machine. Using CRISPR-Cas9, what was the primary target of that initial genetic overhaul?
So the first step was focused purely on offense. It was about eliminating the primary immediate triggers of what we call hyperacute rejection.
The instant rejection.
The instant rejection. These are sugar molecules or antigens that are found only on pig cells. The human immune system sees them, recognizes them instantly, and attacks.
So they had to make the pig heart look less big.
Precisely. The scientists performed what is called a triple knockout, or a TKO. They removed 3 key genes that are responsible for producing those major rejection targets.
Yeah.
Alpha-gal, New5GC, and the STA epitopes.
Okay, so if they successfully knocked out the 3 main hyperacute rejection triggers, why did rejection happen at all, even after 2 months? What mechanism did the genes not fix?
Right, and that's where the other 7 modifications come in. You see, knocking out foreign antigens is only half the battle.
That's the offense.
That's the offense. The pig was also modified by adding human genes. We call them transgenes. And these were designed to play defense against the human innate immune system.
So putting a human shield on the pig's heart.
In a way, yes. They engineered the pig heart to express human complement regulatory proteins, or CPRPs, things like CD46, CD55, CD59. Their job is to stop the complement system from kicking off this generalized inflammatory cascade.
And there was one more, right, for blood clotting.
Just as critically, yes. They added human thrombomodulin, or HTBM, to prevent coagulation pathway dysregulation. That's a huge factor in delayed graft failure.
So that whole package really sets the stage for the short-term success. Let's look at the patient's course. David Bennett Sr. survived 60 days. But beyond the obvious breakthrough, there were some early clinical signs that, looking back, were pretty concerning.
There were. He survived the early period. He even recovered from an abdominal infection that required some really strong immunosuppressants. But the physicians noted something unusual about the heart itself.
What was that?
It was beating with excessive force and speed. They actually needed specific medication just to try and relax its activity.
Wow.
That was a sign, really, that the organ was struggling to integrate long before the ultimate failure.
And his death after 2 months, it demanded this immediate forensic autopsy. The failure, it wasn't a single clean event. What were the 3 main suspects in the postmortem analysis?
The investigation really centered on 3 things. First, had the patient generated a new wave of anti-pig antibodies?
Sure.
Second, were anti-pig antibodies unintentionally introduced through medical treatment?
Introduce, okay.
And third, and this is perhaps the most critical part, was there endothelial injury in the graft that was linked to the activation of a pig virus called Porcine Cytomegalovirus, or PCMV, right inside the heart tissue.
Okay, let's focus on that microbiological risk, the PCMV. We often hear about retroviruses like PERV in this context. Why did PCMV activation become the immediate, the alarming focus after this transplant?
Because PCMV is highly detrimental and potentially an immediate killer. I mean, PERV is a long-term sort of theoretical risk, but PCMV activation, we know from preclinical baboon models, it significantly reduces cardiac xenograft survival.
And in this patient?
In the Maryland patient, it's very likely the latent PCMV was activated because of the intense immunosuppression he needed to prevent T-cell rejection. And that activation just caused massive inflammation and damage to the heart's endothelial lining.
So the source pig, even with all those modifications, it must have been carrying a latent version of this virus. What does that mean for safety protocols for donor pigs now?
It means the bar for viral safety has been raised, dramatically. It is now absolutely mandatory for all donor pigs to be maintained in what are called designated pathogen-free or DPF facilities.
And that's not all, right?
No. On top of that, before any transplantation, they have to confirm the absence of PCMV using highly sensitive PCR assays and serological methods. This adds a critical, very time-consuming layer to safety screening that just, it wasn't sufficient before.
So we've established PCMV as the immediate killer. But what about the slow burn threat, the retroviruses like porcine endogenous retrovirus or PRV? Why did the FDA prioritize the fight against PCMV and basically accept that complete inactivation of PRVs was not required for this initial study?
Well, the regulatory body, they recognize the immediate terminal urgency of the patient and the desperate need for xenotransplantation. While PRV remains a major area of research, it's a virus literally integrated into the pig's own genome, the consensus, and the FDA accepted that the immediate danger posed by known zoonotic pathogens like PCMV was the higher priority.
Right.
You have to solve the problem that kills the patient in 60 days before you worry about the one that might affect them in 60 years.
That makes sense. Okay, let's shift focus to the patient's immune management. The protocol used an anti-CD40 antibody, KPL-404. What does that specific immune agent do, and why is it considered like the backbone of modern xenotransplantation?
Okay, so think of T cells, the core immune assassins, as needing a kind of two-factor authentication before they launch an attack. This anti-CD40 antibody protocol, it's designed to be highly effective because it blocks that co-stimulation pathway, specifically the CD40-CD154 interaction.
It blocks the second factor.
Exactly. By blocking that second signal, you essentially prevent the T cells from issuing the kill order. And that's critical for preventing delayed rejection. This strategy has been the key to achieving the longest xenograft survivals we've seen, up to 9 months, in our baboon models.
It sounds like a perfect defense strategy, but rejection still happened. And this brings us to that challenge of exogenous treatments, specifically intravenous immunoglobulin or IVIG. Why did giving the patient a routine, necessary blood product present such a unique immunological risk?
Because IVIG, which is sourced from thousands of pooled human plasma donations, carries this explicit, massive risk of inadvertently infusing the recipient with anti-pig antibodies.
Wow, so it's already in the general population.
It is. Even in the general human population, there are circulating antibodies against pig antigens. So unless that IVIG product is specifically adsorbed, which means it's filtered against pig cells or a donor phenotype organ, before you infuse it, you are actively introducing rejection agents into the very recipient you are so desperately trying to save.
That suggests a terrible feedback loop. The sicker the patient becomes, the more likely they are to need these therapies, and the more likely those therapies are to accelerate the rejection process.
It is a profound clinical challenge. It truly is. And to make things even more complicated for the research, even our primary preclinical model, the pig-to-baboon model, is flawed.
How so?
Well, we know that baboons, because they're old world monkeys, they actually have natural antibodies against one of the pig antigens we tried to knock out, New5GC. So when we use these TKO pigs in baboons, it unmasks a different, still unidentified antigen we've nicknamed the fourth xenoantigen.
So testing in baboons is like using the wrong color filter on a camera. The results look distorted. They show a rejection mechanism that simply doesn't exist in humans.
That's a great analogy.
Because humans don't have that natural antibody against this fourth xenoantigen.
Exactly. This whole phenomenon, it complicates our modeling. And it probably overestimates the barriers to success in humans. But the fundamental scientific conclusion, it still remains. Deleting new 5GC is absolutely necessary for reducing antigenicity to human serum, even if the non-human primate testing hurdle looks artificially high.
Okay, so if we take all these hard-won lessons, the viral contamination risk, the sort of poison pill of IVIG, the flaws in the baboon model, how are they shaping the future protocols and, I guess, the genetic engineering of the next generation of organs?
They're shaping it in every possible way, including size. We saw a major long-term issue in animal models. The pig heart would eventually overgrow.
It would just keep growing.
It would keep growing, causing detrimental diastolic pump failure. The pig heart, driven by pig growth factors, just kept getting too big for the recipient's body.
So what was the genetic solution for that? To stop an organ designed for a much larger animal from just growing indefinitely?
Researchers created pigs with what's called a growth hormone receptor knockout, or a GHRKO gene. This one specific modification has successfully reduced the organ's weight by about 50%.
That's a huge difference.
It is. And critically, it has facilitated much longer survival, extending well beyond 6 months in baboons. It confirms the role of these intrinsic growth factors in long-term failure, and it gives us a way to genetically control the organ's size.
So looking at the immediate future, what's the minimal genetic modification set that researchers now believe is required to go back into the clinic?
The field is really starting to coalesce around a defined set that tackles both the offense and the defense. That's the inactivation of the 3 primary sugar antigens, plus the transgenic expression of the human complement and regulatory proteins, and the human thrombomodulin. That's considered the minimal viable set to overcome the most immediate immunological and coagulation barriers.
And what's the new success benchmark? How long do these xenografts have to consistently survive in primates before the scientific community feels confident enough to warrant moving to the next level of human trials?
The new benchmark is consistent orthotopic survival, which means the organ is in the correct anatomical position and it's functioning of non-human primates for up to 6 months. And that has to be achieved without irreversible rejection or uncontrolled infection.
6 months.
6 months is believed to be sufficient to warrant moving toward clinical trials in very carefully selected patient populations.
And who are those carefully selected patients going to be? They're not going to be low-risk patients, are they?
No, absolutely not. Future trials should initially target patients for whom a human allograft is already highly unlikely. So this means high-risk elderly patients, or perhaps most importantly, highly sensitized patients with a high panel reactive antibody.
Meaning they react to a lot of human donor tissues.
A lot of them, yes, but who fortunately lack pre-existing anti-pig antibodies. For them, xenotransplantation offers a unique and immediate path forward.
This deep dive has really shown us that the 60-day survival wasn't the finish line. It was maybe the most crucial piece of primary research we've had in decades. The initial success proved feasibility, but the failure that illuminated these specific controllable hurdles.
Absolutely. The forensic analysis, it pinpointed manageable factors. Viral safety, which now demands DPF protocols for PCMV. And the careful balancing of powerful immunosuppression versus the potential antibody introduction from routine therapies like IVIG. These are, they're challenging biological management issues, but they are solvable.
They're engineering problems, not fundamental roadblocks.
Exactly. Solvable through technology and improved clinical protocols.
So as you reflect on all this material, what's the final provocative thought we should leave our listener with regarding the future of xenotransplantation?
Consider the regulatory tension here. The current evidence suggests that getting sensitized to a pig xenograft is not believed to be detrimental to a patient later receiving a human allotransplantation. This makes a really compelling clinical argument for using xenografts as a critical bridge for high-risk patients who desperately need a temporary solution, especially infants.
A bridge to a human heart.
A bridge to a human heart. But, and it's a big but, the heavy immunosuppression required to keep that xenograft alive risks activating latent viruses like PCMV, which could pose a new infection threat to the public health system. So what is this tension? The potential for life-saving immunological freedom versus the risk of a novel infection? What does that mean for large-scale regulatory approval and manufacturing? That is the balance that defines the entire future of this field.